Hi, I'm Jeanine Ash from Rice University in Houston, Texas, where I am a Center for Dark Energy Biosphere postdoctoral investigator. The largest reservoir of methane on Earth is actually below the seafloor in sediments. Archaea, called anaerobic methane oxidizers, or ANME for short, work with microbial partners to eat that methane and keep it from getting released to our atmosphere. That's methane, we hope. I work on understanding this really delicate balance between the methane that's below the seafloor and the microbes that keep it out of our atmosphere. And really, most importantly for this expedition, how sensitive are they to changes in the ocean temperature and nutrient loading that's happening due to anthropogenic climate change?
And as soon as these cores are cut open, we stick in these sterile syringes in order to take our samples. All right, and once both syringes are in, pull them out really gently so that the sediment comes as well. Set them in that nice clean foil. All right, and we're done on the catwalk. And now I'm going to subsample as quickly as possible. So the first thing that I'm going to do is I'm going to take my syringe, and 1 cc of it goes inside this test tube, and I cover the top of it with formaldehyde. So one of the questions we want to know is actually how many. We want to quantify the microbes that are down there. So this formaldehyde is going to kind of fix the cells and preserve them. And then the second one, this is the whole sample, so I get rid of the plunger like that. This one I'm going to measure metabolomics on, and so that's sort of like all the tiny small molecules. They could be both organic or inorganic that microbes make and microbes use. And then come over here, and 5 C's goes into this test tube, and we store it with a solution called RNA later. This is for microbiologist Shiv, and this solution is going to help preserve little bits of DNA and RNA. Another 5 cc's gets dropped into this bag. And so he's going to flush that with nitrogen so we can store it in as oxygen-free of an atmosphere as possible. And that special packet he's got is also going to help suck up any oxygen that gets left over there. And then it goes inside that wine bag that he can seal. And then this final sample of his is one that is actually for fungal cultures, and it just goes in the bag by itself. And these all get stored once they've been treated this way in a variety of manners. So some of them go directly into a -80 C freezer, others get stored into a -20 freezer, and then others get stored at 4 degrees. And that's microbiome on the JOIDES Resolution.